So this is today is more of a tutorial. So you'll have an exam, a test this Wednesday at 2 p.m. So any questions about? So the test will be based on your practical schedule. There will be 3 questions. You'll be asked to carry out some calculations and yeah, analyzing data and all that. You have any questions about the practical that you'll do not understand?
No, doctor.
So maybe I'll just I'll just point out some of y'all have submitted the assignments, so I'll just point out some of the common mistakes, okay? Can you see the PowerPoint slide, practical one?
Yes.
Okay, so this is very straightforward. Calculation of TSS and POM. So TSS is total suspended solids, so it's W2 minus W1. Okay, divided. So if you look at the unit here, it's milligram per liter. So this is gram. So of course, then the counts will be like 5,000. Very high numbers. Okay. So you take note, but I think I think this was a mistake. It's supposed to be in milligram. But anyway, you make sure the units are equivalent, then per liter. Since this is from 500 ml filtered sample, that means it's a 0.5 liter. So you have to divide your difference here by 0.5. Okay, that will give you your total suspended solids. For particulate organic matter, it is this filter that's ashed, the placed in the furnace to give you this uh, weight. Okay, so this minus this, instead of since particulate organic matter, you're looking at the weight loss upon on ignition. So how much organic matter will be burned off when you place it in a oven at 500 degrees Celsius for 2 to 3 hours. So you are looking at the weight loss to represent POM. So the weight loss will be this minus this. Okay, that will give you your, then divided by 0.5. That will give you your POM. Okay, so very straightforward. Then similarly for practical 2, then for this you have to determine from uh, one sample t-test against the criteria of 50 milligram per liter and 5 milligram per liter of TSS and POM to determine whether the measurements that you obtained were within the water quality limits. So any questions for practical 1? Any questions?
No question, Doctor.
Thank you. Are you all having your biosymposium next week?
Yes, with
Okay.
13.
Okay. So for practical 2, this is just how inorganic nutrients are measured in marine chemistry. First, a standard curve is prepared for the different measurements: phosphorus, ammonium, and silicate. So what you have to do is you have to draw a standard curve, which is basically a linear regression curve. Then from the equation, convert this absorbance reading into micromolar. Once you have this, okay, so these are from 5 stations within the same lake. Then you want to determine whether the concentration from this lake is within standards. Okay, so you do one sample t-test. Okay, so all these 5, you use all these 5 measurements here, and then test against the criteria to determine whether it's within or outside the standard. So this is quite straightforward. Yeah, but for those who haven't done it, please try it. Please try it at least once. Or if you know, if you've seen the video, then and you know that you can do it, then it's okay. But you should try it because yeah, if you're doing it for the first time, it might not be uh, you might not be able to do it fast enough. Okay. So any questions for practical 2?
No, Doctor.
Okay. So phosphorus is important for primary producers, phytoplankton. Okay, example. Ammonium, also important for primary producers. Ammonium is also quite interesting because it is usually high when the level of dissolved oxygen is low. Silicate is important only particularly for diatoms. Usually when we measure inorganic nutrients, we will also measure nitrate, NO3. So that will give you a good idea of the level of eutrophication in your waters in your waters. Also give you an idea of whether it's N limiting or P limiting in your waters. Okay, so now we're looking at practical 3, determination of primary producers. So for this, the primary concern is how to use the formula here. Okay, so if you look here, it's for chlorophyll A, these are all straightforward. You see a small v and a large V here. So the small v is volume of acetone in mil. Then the large V is volume of seawater in liters. So if it's already 0.5 liters or if it's in 8 mil, you don't have to convert everything to liters. Okay, because the equation has already taken into account the different units. Okay? So that is make sure you do not convert unnecessarily, because look at the instructions carefully and you know that this one is 8 and this is 0.5. Okay, so the next is this one, 6650. So 6650 is actually the absorbance at this minus this. So you have to take this minus this and then put it here. Okay, put it here, 6650. Then for 6650A, it is this minus this, then you put it here. Okay, so it is not direct not directly used, but you have to minus off the 750 first. So as I've mentioned before, 750 is to account for the different turbidity that may occur in your samples. So yeah, so this is how you use the formula. This is a monochromatic equation to determine the chlorophyll A pigment concentration. Next, for diversity index, okay, if you want to do it, you can do it manually using Excel, but you have to type your formula in one by one. So this is a summation. So each genus have to be calculated. So the example for station 1 is shown here. So 8 over 11, ln 8 over 11 plus 1 over 11, ln 1 over 11, 2 over 11, ln 2 over 11. Okay. If you use past, there is a function there that allows you to calculate for Shannon's diversity index. So you just need to copy and paste and arrange it in a manner that the software can understand, and then it will give you the output immediately. So from here, probably you can see that among the 5 stations there are differences, especially when you have high ketoacidosis. Next is the light dark dissolved oxygen method. So very straightforward. You just need to know the slope. So the slope you can calculate by using a linear regression equation. Okay. Then from the slope, what you see in the light bottle will be your NPP, net primary production. What you see in the dark bottle is your community respiration rates. And then the difference between this and this will give you your gross primary production. So if you notice, the slope here is negative and the slope here is positive. Okay, but you want the difference between the magnitude of the slopes. Okay, so it has to be this slope minus this slope, so you actually a double negative will become positive, so this will be this plus this. Okay. Any questions about practical 3?
No question,
No
doctor.
question, doctor.
Okay. So for practical 4, is this just a way of using the software to to process your flow cytometry data in order to determine the different fractions, okay, based on color and based on size. So for this example, I've given you the flowing software, an example of flowing software, but this is only available for Windows. Rehana, I remember you use Mac, so how do you do this?
I have 2 operating systems on my laptop, so I have to shut down my OS, go into
Okay.
Windows, and then.
Oh, so you are able to do it. Okay, good.
Yeah.
So there is a Mac version, not a Mac version for this, but a different one that uses Java based. But that one is still in development and not so straightforward to use. Okay, luckily everyone can use the flowing software. So, but as I've mentioned before, depending on the type of if you're using it in your own research or in your work, all equipments will have their own proprietary software that you can use to analyze. Okay, the most important thing is to understand uh, the use of forward scatter, side scatter, and then the use of the availability of different channels. So the capability of a flow cytometer is dependent on how many channels it has. A low-end flow cytometer will have about two channels, so that will cost maybe about 80 to 100,000 ringgit. Whereas a high-end can easily go up to 500,000, and that one can have 6 channels. 6 channels means uh 6 channels means you have different lasers and different filters, so you can cover a lot of different colors. So you can use more fluorochromes, different kind of fluorochromes. You can do more separation, and the highest end will always also have a cell sorter, which means you can actually sort your cells. You can actually collect the different cell fractions that you want. Okay, so depending on the uh, price of the flow cytometer, you have different capabilities. Okay, any questions about practical 3? Practical, this is practical 4 already. Okay, okay, no questions on practical. So practical 5. Okay, so this is it's just to show how size fractionation is used in microbial ecology for marine work, where when you filter certain fractions, you can assume them to have to not to have any grazers or predators or protozoas inside. So in the less than 0.7 micron fraction, there are no predators. Then in the unfiltered fraction, unfiltered seawater, there are predators. So the difference between them will indicate the action of your grazers, your protozoa. So a common question is, what about viruses? You know there are viral lysis. Okay, so why will viral lysis affect this kind of a setup?
Yes.
Um,
What's the question again? Sorry.
will viral lysis affect this setup?
Yeah, I think so because virus is very small and it actually can pass through it. So
But they are available in both fractions.
yeah, true. Yeah, so it's a
So we
constant.
can uh this one is not really tested. So theoretically, it shouldn't be a big problem because virus is available in both fractions. But the problem is viral activity is dependent on other components. So because other components also change, there might be differences in viral activity. Okay, but usually we do not consider it unless you are studying viral lysis. So probably you can explain that since viral fraction is available in both, so we can ignore them. Okay. So the main differences between the two fractions will be the action of protozoa and predators on the bacterial population. Okay. So usually in my lab, because we do not focus on specific bacteria, we will use DAPI. We will use direct count to look at how bacteria change, how bacteria is grazed by the predators in the total fraction versus the filtered fraction where supposedly there are no grazers. So the difference will tell us the grazing rate that is present in the sample. Okay. However, for class practicals, usually we inoculate E. coli inside because E. coli is easier to detect. Okay, and it is also quite interesting because a lot of our wastewater, although treated, still has some E. coli in it. So what happens when the E. coli is discharged into the river? What happens to them? So their E. coli is a freshwater bacteria. So, but they are also being grazed once they enter the natural river system. So therefore, we can estimate the die-off rate, the half-life of E. coli in the environment. Just remember that the half-life of E. coli in fresh water and seawater is very different because E. coli doesn't survive well in seawater. Okay, they survive better in freshwater. There are a lot of there are a lot of factors to consider because E. coli can also attach to soil. They can also go to the benthic environment. So a lot of things to consider if you want to study uh, the presence of E. coli in water systems. So here, very straightforward. This is a replication of 5 replicates of the same water sample. Here is the total seawater here after 24 hours. So these are all inoculated E. coli. Here is the size. Here is in the seawater, but less than 0.7 fraction. So from here, why do you think there are more E. coli here than here? Although we inoculated the same amount at 0 hour. Any reason? I mean, we inoculated the same amount of E. coli in your samples. So, but why is it higher here than here? I mean, a little bit higher, but still, you can see that the order is different.
Maybe some of the greatest being calculated.
Okay, some possible reason is this is total. That means maybe it also has some E. coli. Maybe it also has some E. coli there already. And also it also have probably a lot more particles. Some particles can have E. coli present attached to it. That's why maybe the numbers are higher here as opposed to here. Okay, that's possible. So we use ANCOVA to compare the slopes. Because ANCOVA compare the differences in the adjusted mean. Yeah, that's why we use ANCOVA to compare the slopes.